Bert got in early that Saturday to open up the store. He'd had his morning coffee in hand as he greeted the clerks and the shelf stackers, got things ready for doors open at 7. It was a fine, late spring Saturday in Davis, California, 1994. As far as Bert was concerned, today would be pretty by the book, with one minor exception. They finally got in their first shipment of the new McGregor tomatoes. These ones weren't like the ones they'd been stocking for the last few months, sent up from those folks at Calgene. Those had just been placeholders, biding time while they waited for the FDA to greenlight the real goal. These were the genetically altered ones. The Flavr Savrs, they called them, but McGregor was the label in the store. And Bert had been sure to put out a little display to let everyone know that these famous tomatoes were finally here. He was a little apprehensive about whether there would be any takers. That price was a little higher than the standard tomatoes, but he had to admit they looked just right, juicy and red, the ideal tomato. By 9 a.m., he knew he shouldn't have worried. They had already restocked twice. He put down his coffee and went back into the back office to call Susan at Calgene and let her know that they'd be doubling next week's order. All through the weeks to come, the Flavr Savr McGregor tomatoes from Calgene were a hit with customers. And not just here in Davis, long a center for American agricultural innovation, but also in the few stores in Chicago that were already selling them, and the stores in San Francisco and LA that were going to get them soon too. Over summer, the demand dropped a little, which made sense. It's hard to compete in the summer, peak tomato season, when juicy fresh tomatoes are half the price of the GMO tomatoes. And maybe the low-level but consistent resistance from Greenpeace and others was having a little bit of an impact. A Boston College professor had recently coined the term Frankenfood in the New York Times, as news was spreading about these biotech foods coming to supermarkets soon, and that word had caught on. Yet after summer, the demand never did really rebound. Maybe the initial demand was more for the novelty, and consumers didn't care enough to put up with the higher price. Especially since the threat of this fancy new lab-grown tomato had put some wind into the plant breeders to put better, conventionally bred varieties onto the market. And maybe it wasn't the best idea for this company that mostly knows biotechnology to take on all of the growing and shipping of these tomatoes. Those are tough things to get right. A few years later in 1997, Bert's store, the State Market in Davis, California, had likely already moved on and stopped stocking the Flavr Savr. And by the end of the year, not just the product, but the entire company that made it, Calgene, were no more. They were bought up and plundered for their core technologies. Their patents were integrated into the growing agricultural biotech company that had started out as a chemicals manufacturer in 1901 in Missouri. It was founded by John Queeny, but for the company he founded, he decided to use the maiden name of his wife, Olga Mendez Monsanto. Welcome to Modified. I'm your host, Dr. Orlando DeLange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 11, the first ever GMO, the Flavr Savr tomato. So between 1994 and 1997, the world had already seen the appearance and disappearance of its first commercial GMO crop on the market. Even though the Flavr Savr itself was doomed to failure as a commercial enterprise, it had a lasting impact in shaping public attitudes to GMO crops, as well as ramping up commercial interest in developing GMO crops. Both of these trends continued at a fast pace through the 1990s and 2000s, despite the individual failure of some companies and products along the way. I intend to create a second and third season of this podcast that will dive into the commercial, social, legal, and other aspects of GMOs, basically getting beyond just the biology and beyond the basic social and historical context. But for now, we'll keep on diving into the biology. What actually was a Flavr Savr tomato? I think a very obvious and easily overlooked starting point is that the parent variety of GMO crops are not built from the ground up in a lab. Essentially, what you're doing when creating a GMO is doing small tweaks to the genetic material of existing crop varieties. While it's hard to track this down, based on my reading of a 2008 paper by Hyatt and Sanders, the Flavr Savr's parent cultivar seemed to be Mountain Spring. That's a popular, widely grown hybrid tomato variety that produces plump red tomatoes, slightly smaller than other tomatoes, but with a reliable yield. I wanted to take a minute to emphasize this because it's easy to think that GMO crop development happens in parallel to other plant breeding and that all the other complex parts of the agriculture industry. It does not. Calgene wanted to make a product that farmers would be willing to grow and consumers would want to buy. All they really had to contribute was a single specific trait, which we'll examine in more detail, but a tomato as a product has many traits. For the farmer, some of those of most interest include the growth habit. Is it trailing or upright? And does it have good pest resistance and disease resistance? Also, how large are the fruits? How many are produced per branch? How susceptible are they to cracking? Do they all ripen together? That's the best for harvesting and shipping. And perhaps most crucially of all, are there existing products, such as harvesting machines and processing machines, that are compatible with the plants that grow from the seeds of this cultivar? When a farmer invests in seeds, whether or not they are GMO seeds, it's like investing in any high-tech product. They're making a sizable financial investment, and there are a whole range of specifications to review. And consumers, of course, have a wide range of expectations. You know this, since you certainly bought or at least have eaten fresh produce. Tomatoes are not all alike. The look, feel, smell, size, shape, and most importantly, taste matter a great deal. Those are features that will not be overlooked. In general, and this is what Calgene did, the company producing the GMO crop will introduce this novel DNA into a set of already existing cultivars. That's to say, they will not just make the GMO once, they'll make it multiple times, and they'll look for the best one. And it doesn't stop there. From the point that they can use all the tools we learned about in the last few episodes to then transfer the transgene, that's the introduced DNA, into existing cultivars through sexual reproduction. So they'll take their GMO lines and cross them with other cultivars, ones that have desirable traits. Let's imagine these parents are G for GMO and D for desirable. Let's imagine that G is the result of adding a transgene into some other existing cultivar that is easy to handle in a laboratory, but doesn't have all the traits that farmers and consumers want. Ideally, you want to take this transgene and then get it into the cultivar that's more desirable. From this point, you can do that using sexual reproduction. Take G, cross it with D, our desirable cultivar, and then use PCR or other techniques to identify the progeny that successfully received the transgene. Then those offspring can be crossed back to the original D parent. These progeny will mostly have DNA from the D, desirable parent, but using PCR you can find those that still have the transgene, that little piece of DNA from the G parent, and then grow them up and cross them back again to that original D parent. Multiple rounds of crossing back to the desirable parental cultivar, but screening to ensure retention of the transgene, is called introgression. It's not just used for transgenes, it's a way to move any single gene or maybe small chunk of several genes into a new cultivar, ideally just those genes and nothing else. In reality, what you end up with is a new cultivar that is about 99% identical to the parent that you're introgressing into, in our case what we called our desirable parent, but has a small amount of DNA from the less desirable parent. But critically, that small amount of DNA includes a transgene or some other allele of a natural gene that is very desirable. This is a way to circumvent the kind of manner in which sexual reproduction totally jumbles the DNA of parents. And instead, you can use this natural process in a very intentional and kind of unnatural way to shift only small bits of DNA between cultivars. So we know that the FlavrSavr was in almost every way a mountain spring tomato. But the almost is important. In addition to all the DNA you'd find in any other mountain spring tomato, it had two genes added. One was a piece of bacterial DNA, and the other was an inverted copy of a naturally occurring tomato gene. The piece of bacterial DNA is just the tDNA from the Agrobacterium tumefaciens, is just a byproduct of introducing that really interesting DNA. The interesting DNA is that inverted DNA that I just mentioned. Specifically, it's an inverted copy of PG, the gene that carries information used to make an enzyme called polygalacturonase. To understand why this would make for a marketable product, we need to know about two things. One, the role of polygalacturonase in fruit ripening, and two, the science of RNA interference, so buckle up. Do you recall from last episode how a young Professor Herrera Estrella in Mexico City was electrified by a talk from a visiting Belgian scientist about the transfer plasmids of Agrobacterium and their potential genetically modified plants? Herrera-Streyer went on to work alongside Belgian professor Jeff Schell to do the first ever demonstration of that principle. And in 1974, British plant scientist Don Grierson watched a talk from Professor Schell and had a similar electrifying moment to that felt by Professor Herrera-Streyer earlier on in Mexico City. Professor Grierson knew right away that he wanted to be part of what seemed like an exciting new field. Remember, this is right around the same time that new tools were being developed by scientists in California, tools to cut and paste DNA. Professor Grierson wondered if he could combine all of these tools, cutting and pasting DNA and the transfer plasmids of Agrobacterium, together with the genes he was studying, genes that control fruit ripening in tomatoes. In particular, he was interested in an enzyme called polygalacturonase. It sounds like a mouthful, but let's break that down. The A's, as the end of an enzyme name, means that it breaks down those things. So proteases break down proteins, and amylases break down amylose, the main component of starch. Polygalacturonase breaks down polygalacturonic acid, one of the main constituents of pectin. Like starch, pectin is a complex carbohydrate. Carbohydrates are sugars, and all the molecules made up of sugars. Sugars are chains or rings of carbon atoms with hydrogen and oxygen, and they are energy rich, so we often think about sugars, and therefore carbohydrates, in terms of their function as energy storage molecules, aka food. But they can also play important structural roles. For instance, the cellulose that gives plant cell walls their strength is also a carbohydrate. And pectin is another part of the plant cell wall. The cell wall is not just cellulose. Pectin plays a particular role at the space where the cell walls of neighboring plant cells meet. It helps to basically glue the cells together. And during ripening, enzymes, notably our friend polygalacturonase, break down the pectin that holds cells together, loosening up the plant tissue. And that is one of the key features of the regulated softening process that we call ripening. Ripening is, though, more than just softening. It often involves a bunch of biochemical changes that lead to modifications in color, scent, and flavor. Ripening evolved to make fruits easier to eat and more attractive to potential fructivores, so plant-eating animals, because the plants that have big fleshy fruits rely on those fruits being eaten to disperse the seeds inside. And of course, during crop domestication, ripening has been a real trait of interest. But it's not something like fruit size, where in general bigger fruits are always better as long as they're still tasty and nutritious. Ripening is tricky, particularly in modern food systems where food is often harvested far from where it is to be eaten, or where fruits might be going into an industrial process rather than directly into a mouth. That's definitely the case with tomatoes on both counts. And so ripening can be tricky. You do want a nice ripe tomato when you eat it, but you don't want a bruised, destroyed tomato because it hasn't survived transport or processing. And so in most cases, the flavor is sacrificed by picking tomatoes unripe so they're sturdy enough to survive shipment and they get to you intact. And then a lot of those tomatoes are sent to be processed into products like tomato paste, and there you actually might want a tougher tomato to do well in the processing. Professor Grierson and his team felt that polygalacturonase could be an ideal case study and a profitable application for this newly conceived idea of using agrobacterium to create genetically modified plants. What if you could somehow switch off the polygalacturonase in tomatoes to slow down ripening or to increase pectin content in the final fruit? How to actually do that? Remember that while today CRISPR offers a way to target a gene inside a genome and directly modify it, that was not possible in the 1980s. Just because you know the gene exists doesn't mean you have a way to sort of go into the plant genome and turn it off. At least not in the 1980s, when Professor Grierson's lab really got to work on this idea. Agrobacterium delivers genes into the plant genome at random positions, so you can't just say target polygalacturonase, or the gene that encodes it. Well, it just so happens that there is actually a process that allows you to shut down expression of a gene in plants, and really in most organisms, as long as you know the DNA sequence of that gene, and that process is RNA silencing or RNA interference. Now, I will spare you most of the molecular details for two reasons. One is that it would take too long, and two is that, as shocking as this may seem, It was never actually clear exactly how this process worked in the case of the FlavrSavr. There are many different mechanisms under the umbrella of RNA silencing or RNA interference, and it can take a lot of research using tools that simply weren't available in the 80s to know exactly which one is taking place in any instance where you observe this phenomenon. Overall, these mechanisms all involve earmarking certain RNAs for destruction based on their sequence, and sometimes involve then earmarking the gene, the DNA encoding that RNA, and adding chemical markers to the DNA that basically tell the cell's machinery, stop reading this gene. But just as there are many mechanisms of RNA silencing, there are several different ways to trigger it. But one of the best is what we call double-stranded RNA. So RNA, if you remember, is the sort of intermediate molecule. It's produced from DNA, and you get a single complementary strand of RNA that comes from reading one of the two DNA strands. And when you have that single-stranded messenger RNA, it's then fed into an amazing molecular machine called a ribosome to produce a corresponding protein based on the sequence of the RNA molecule, which, if you remember, comes directly from the sequence of the DNA molecule. But anyway, just because RNA is normally one strand, it can, just like DNA, form a double-stranded molecule with two strands joined together based on complementary base pairing, because at the molecular level, it is very similar to DNA. And when this happens, when a double-stranded RNA molecule is formed, this is recognized by specific cellular machinery that triggers RNA silencing of that RNA molecule and any others that have the same sequence. To make an RNA double-stranded, you don't want two identical RNA molecules, otherwise all RNAs would end up double-stranded. When genes are expressed, this is happening at scale. So remember, inside any given genome, a gene that's expressed is being constantly recognized by specific cellular machinery that initiates the process called transcription, which churns out many, many copies of RNA from that gene. So at any given time, you'll have many basically identical RNA molecules coming from each gene, that is, at least each gene that's being expressed. Sometimes thousands of identical messenger RNA molecules being produced per hour from a given gene. So if those identical molecules, they cannot bind to each other because this base pairing process of both DNA and RNA requires complementary sequence, not identical sequence. So to get a complementary sequence, there is a fairly simple way to do this. If you know the gene that you're kind of interested in, you can easily work out, through the simple rules of complementary base pairing, a reverse complementary sequence of the original gene. And then if you put that into a cell and have that expressed, the RNA produced from that gene will have the reverse complementary sequence of the original RNA. And if those two RNAs meet each other, they can come together and form a perfect complement and a double-stranded RNA. Strangely, that idea is something that two groups were doing at the exact same time in the mid-1980s. So one in the UK we just heard of, Professor Grierson's lab, and the other in Davis, California. Those were the scientists at Calgene, the world's first plant genetic engineering company founded by Ray Valentine in 1980, who we've already heard a bit about. Professor Grierson wrote an article about the history of his lab's work at this time, and I've linked it in the show notes. And as far as I can see, it was a total coincidence that both teams took this exact same approach. That's both interest in a gene called polygalacturonase in tomatoes and using RNA interference to reduce expression of that gene. Seems a little odd, that's quite a coincidence, and if any of you listening know more about what really happened there, do let me know. At any rate, in the mid-1980s, teams across the Atlantic took the gene that encodes the tomato polygalacturonase gene, synthesized a coffee with the reverse complementary sequence, and used the agrobacterium tDNA method to insert it into the tomato genome. And the consequences are what I described in the cold open. There is a twist in this story, which connected some dots for me as I did my research for this episode. And as you can tell, I grew up in the UK, not the USA, and I also recall in the early 90s lots of controversy around GMO tomatoes, but the FlavrSavr never made it to the UK. That's because there was in fact a separate GMO tomato product almost exactly identical to the FlavrSavr developed and marketed in the UK by a different company, in this case Zeneca, working with Professor Grierson and the technology developed in his lab. That is the same Zeneca that today is part of AstraZeneca, the drug company. In 2000, the agricultural wing of Zeneca merged with the agriculture wing of Swiss drug company Novartis to form a new company, Syngenta, which to this day is a major producer of GMO crop seed, among other products. Not to get too far off field with this tangent, but Syngenta is headquartered in Switzerland. And in neighbouring Germany, you can find the headquarters of Bayer. And those who track the comings and goings of the agrotech world will know that in 2018, the world's GMO boogeyman, Monsanto, was acquired by Bayer. So we have Syngenta in Switzerland, Bayer in Germany, and yet Switzerland and Germany both do not allow GMO crop cultivation. These are the sorts of fun twists and turns that will be focused on more in Season 2 of Modified. But anyway, Zeneca released their product in 1996, beaten to the chase by 2 years by Calgene. And that's why Calgene and the FlavrSavr tomato still have that accolade of first ever commercial GMO product. Also, Zeneca's product didn't have a flashy brand name like FlavrSavr, and that's because the tomato itself wasn't something you could pick up in the produce aisle, where a specific brand name is important for customer recognition. Instead, Zeneca's GMO tomato graced the canned goods aisle in the form of tomato paste. A tomato paste that was significantly cheaper than its rivals, and was actually, unlike the FlavrSavr, a big commercial success. The Zeneca GMO tomato paste went off the shelves too, however, just a couple years after the FlavrSavr, but in this case it was due to large-scale opposition to GMO crops that started to form in Europe at just the same time as these products came onto the market, and that grew and grew through the 90s and 2000s. And yes, not to be too much of a broken record, but that is indeed something we'll look at more in Season 2. So if the Zeneca tomato failed due to a consumer backlash around the concept of GMOs, why did the FlavrSavr fail? I'll explain this with a pinch of salt because it comes from an article written by Professor Grierson, and there was clearly some animosity between his team and the folks at Calgene in the US. To understand why the FlavrSavr failed, let me explain more clearly what the concept behind the product was. So remember, the tomato cultivar produces less of this enzyme that breaks down the cell walls in ripening fruit because they've knocked down the expression of the gene polygalacturonase that breaks down pectin during ripening. But it doesn't stop the other processes of ripening. So this is a key thing to think about, is that this fruit, it would be essentially firmer, but it would still develop color and would accumulate sugars and other molecules that we think of when we imagine a ripe tomato. So basically it would be a tomato that just stays relatively firm as it ripens. Another thing to keep in mind is that in contemporary food systems, our food generally has to come on a long journey before it gets to us. Food needs to be transportable, and traditionally, ripe tomatoes are not that. They would simply turn to mush before they even got to a grocery store. So tomatoes, like many other fruits, are picked unripe and then put into boxes. Think about that. Packing thousands and thousands of tomatoes from one store into boxes as quickly and efficiently as possible, that is not going to be a gentle process. Those fruits need to be pretty tough, and when they get to a distribution site nearer to the grocery store, they are then exposed to ethylene gas to trigger ripening. That might sound bizarre, but ethylene is a simple molecule that is naturally produced by plants to trigger ripening. It's one of the major plant hormones like our friends auxin and gibberellin. It is easy to chemically synthesize, and it's been used for this purpose, artificial fruit ripening, for decades. And if anyone's really interested in what I just covered there in 2 sentences, let me know, and I'll be happy to dive into it in more detail in the upcoming Your Questions Answered episode. So the fruits are artificially made to ripen before getting to the store. And that more or less works, but not perfectly. Ripening on the plant just works better, for a number of reasons. It gives you a more flavorful fruit, for one. For tomatoes, just as much as for other fruits. So the exciting idea of the FlavrSavr is that it could ripen on the vine, but still be solid enough to survive packaging and transport. The flavor would be saved. Or maybe we could all savor the flavor. One of those. Something about flavors, saving, savoring, you get the idea of where they got the name from. I think a lot of people could get behind that idea of essentially a more flavorful fruit. And indeed, from what I've read, in general, members of the public liked the idea, hence the interest and enthusiasm that accompanied its launch in 1994. However, that great idea was not actually so great in practice. These GMO tomatoes were indeed harder than normal tomatoes, but not quite hard enough for another aspect of modern tomato farming, which is mechanical harvesting. Most tomatoes are not picked by hand, but by a large machine driven slowly across the field, which rips up the vines and shakes the fruits free. The ripe Flavr Savrs just could not actually survive that. What I can't pin down is whether that meant that a lot of them got crushed during the process, raising the overall production costs because you lost a lot of your material on the way, or that they had to be picked by hand, which, as you could imagine, would massively raise production costs. Either way, it meant that they were significantly more expensive. And consumers did enjoy the Flavr Savrs, they liked them overall as a product, but they just didn't find them worth the extra cost. And so after the initial novelty wore off, the shelves started to pile up with unsold produce, and Calgene were in trouble. And so who would come to their rescue but the world's most famous plant biotechnology company, Monsanto? Like other companies already mentioned in this episode, Monsanto are a relatively old chemicals manufacturer, who in the mid-20th century were part of the growing technoscientific transformation of American agriculture, and they started making agrochemicals like pesticides and herbicides. Monsanto, like many others at the time, saw that biotechnology was going to be a big deal and they wanted in on the action. They started their own department to create genetically modified plants in the early 80s, and they released their first GMO product, the Roundup Ready soybean, in 1996. Just a few years after the Flavr Savr tomato was launched. And we'll get more familiar with that particular product, the Roundup Ready soybean, in our next episode. But just to say, while they may have been beaten to the accolade of first ever commercial GMO by the smaller, pluckier Calgene, they were definitely in the race, and they didn't let that setback get them down. They simply bought Calgene, acquiring their patents and their scientists just as the Flavr Savr was floundering. That was in 1996, and a year later, the Flavr Savr was no more. From what I can find out, it doesn't seem that Monsanto bought Calgene in order to kill off the Flavr Savr. Rather, the Flavr Savr was already struggling, and they maybe just saw it wasn't worth keeping it going. Meanwhile, in 1997, the Zeneca GMO tomato paste was still going strong in the UK, and that's because they pursued a different model for what was otherwise a very similar product. Instead of trying to get vine-ripened tomatoes into stores, they worked out that these tomatoes actually worked better for making a tomato paste. For tomato paste, you want a sturdier fruit, it just gives you a better paste. And so normally, during processing, the harvested tomatoes are heated up to destroy the polygalacturinose enzyme and preserve the cell wall structure of the fruit, essentially achieving, through an application of heat, what has been achieved through genetic engineering. But that heating costs money because it uses up energy, and it also damages the flavor of the eventual paste. So what Zeneca found was that they could produce a paste that was cheaper and also with a slightly better flavor. And it was a hit while it lasted, before the opposition to GMOs as a concept grew too much for the product to be viable anymore. The story of the Flavr Savr holds so many of the themes that come up again and again when we think about GMOs. The exciting scientific idea that meets the realities of profit and loss. The disconnect between what qualities consumers might want from their food and the requirements of the technoscientific systems required to produce vast quantities of food at scale and deliver it anywhere at any time. And of course, the massive agrochemicals companies stomping onto the field where they remain the biggest, but certainly not the only players in the world of GMO crops. We'll put a pin in all of those themes and pick them up in our next episode, which will be all about the big two traits when it comes to GMO crops, Roundup Ready and BT. But for now, we'll wrap up with our 5 take-home messages. Number 1. The Flavr Savr tomato was launched for sale in 1994, the first commercially available GMO crop. Number 2. The Flavr Savr was pulled from the market in 1997 after disappointing sales and failing to make a profit. Number 3. The Flavr Savr was created by adding in a reverse complement version of the polygalacturinose gene, which reduced expression of the native version of the gene via a process known as RNA interference. Number 4. The same approach was used to create a GMO tomato in a UK-Swiss venture. This was successful at first, but fell to pressure from an anti-GMO backlash. Number 5. The Flavr Savr was created by Calgene, but they were acquired in 1996 by Monsanto, a much older and larger company. Okay, that's it for episode 11. Episode 13 is going to be another Your Questions Answer episode, so please get me your questions to orlando@modifiedpod.com, that's ORLANDO@modifiedpod.com, or at Blue Sky @OrlandoDL for Delange, or Instagram, that's @SeattlePlantNerd. And if you didn't catch that, you can find all of my details at the website for the podcast, which is modifiedpod.com. As always, please do subscribe, leave a review, and tell a friend. The number of listeners is growing slowly, but I would really love to reach a broader audience, and if you can help me do that, then well, that would be great. Bye!